Hello, everyone. Welcome back to the fourth and final portion of the digestive system. Thankfully, this one is going to be very short, especially compared to part 3. So here we're going to be talking about the various accessory digestive organs, including liver, gallbladder, pancreas, and what's called the biliary apparatus. So the liver's job as far as the digestive system goes is for the production of bile, which is very important for breakdown of fats. Bile is then stored in the gallbladder, and bile is going to get from the liver and the gallbladder into the intestines via the biliary apparatus. Along for the ride here, we have the pancreas. We talked about the pancreas in the endocrine system, but that was only 1% of the pancreas dealt with the endocrine system. 99% of the pancreas plays a digestive, an exocrine portion. This is what's going to make all the various digestive enzymes, or most of the digestive enzymes that are actually going to break down our food. So those enzymes are produced in the pancreas, and they're going to be transported to the duodenum. Okay, so basic structure of the liver, we have 4 lobes: right lobe, left lobe. Easy enough. And we have the caudate and the quadrate lobe. So if we go to the picture, right lobe, that's the big one. Left lobe is a smaller one. Then we have the quadrate lobe and the caudate lobe. Quadrate and caudate. One way to keep these separate: caudate sounds like caudal, right? Remember, caudal means towards your tail, so it's more posterior. So the caudate lobe is on the posterior side of the liver. Here we have the gallbladder kind of tucked under the liver. And yes, the gallbladder is actually green because bile is green, right? Pretty obvious to see. We don't get a chance, unfortunately, to do dissections right now, but normally when you open up an animal, you will definitely see a little bright green little bladder there, the gallbladder. We have the, let me get my pointer going here. We have the inferior vena cava passes through this structure. This taken together, these 3 structures are called the porta hepatis, right? Portal into the liver, or doorway into the liver. So they're made of the common hepatic artery. We have the hepatic portal veins, and then we have the cystic duct, or a common bile duct right here. So we'll get into that in a second. The common hepatic artery is bringing nutrient poor but oxygen rich blood from the left side of the heart. The hepatic portal vein is bringing oxygen poor but nutrient rich blood from the intestines to the liver in order to be cleaned. The green tube here, the common hepatic duct, right? This one right here is a common hepatic duct, not this one, that's different. This one is the common hepatic duct, and it's draining bile out of the liver. So bile is produced in the liver, transported down the common hepatic duct, stored in the gallbladder, and then can then make its way eventually into the duodenum. But there are lots of little structures here we'll take a look at the very last slide in this lecture. So hepatic veins drain into the inferior vena cava, that's out. The hepatic artery brings oxygen to the liver, so that's in. The common the common portal vein brings nutrient-rich blood into the liver. That's the weird thing about the portal veins, if you remember that from our circulatory system. And the common hepatic duct drains bile out of the liver. Now, these other 2 things here, round ligament of the liver and the ligamentum venosum, for our purposes, these are just structures you need to be aware of. Where these things actually come from, this is a leftover of fetal development. Remember, we talked about the heart. We looked at the ductus arteriosus of the ligamentum arteriosum, right? This was a leftover. This used to be a blood vessel connecting the pulmonary trunk with the aorta. These 2 things right here are the same concept. We had some bypasses. Early on in development, fetal liver is not working, so it gets bypassed. So the round ligament of the liver used to be the umbilical vein, believe it or not. And the ligamentum venosum used to be the ductus venosus. This was basically a way to bypass the liver. So blood was coming in from the placenta, and it's going to go through your belly button, go through your navel, and it's going to bypass the liver mostly. And that's why we have these two structures. When you're born, they cut the umbilical cord. The stuff is lost, right? You don't need, these vessels go nowhere. So they kind of atrophy and they go away and they turn into these two ligaments right here. So you don't need to know the story of fetal development, but you do need to know these structures. Okay. Right. Basic anatomy, I just covered this, but understand you're going to find the inferior vena cava. This is going to be in the posterior side of the liver. You're going to have that ligamentum venosum, leftover fetal development, gallbladder, round ligament, also a leftover of fetal development, and you'll have porta hepatis, how oxygen-rich blood and how nutrient-rich blood get into the liver and how bile gets out of the liver. So here is the histology, a cartoon version of the liver. You take the liver, make a blow up. Liver is subdivided into structures called lobules. So this is one lobule. So they are six-sided hexagonal structures, 1, 2, 3, 4, 5, 6 sides. In the center of a lobule, you'll have a central vein. Central vein eventually connects to the hepatic veins. So this is how you get out of the liver. At each corner, at each vertex, you have what's called a hepatic triad. So a hepatic triad are 3 tubes. So if we take a zoom in here, right, this is one little section out of one lobule. There is the central vein. Here is our hepatic triad. The hepatic triad is made of 3 structures. A branch of the hepatic artery, bringing oxygen-rich, nutrient-poor blood to the liver, so liver cells can get their oxygen. A branch of the hepatic portal vein. This is draining oxygen-poor, nutrient-rich blood from the intestines, the stuff that needs to get cleaned in the liver. And you have a bile ductal. So a bile ductal, this is draining bile out of the liver. So central vein is out, hepatic artery is in, hepatic portal vein is in, bile ductal is out. So the way this all works together is branch of the hepatic artery, branch of the hepatic portal vein. These two structures are going to merge and you're going to mix the blood. So now you're going to get purple blood because it's not really blue, it's not really red, it's not really clean, it's not really dirty, it's both, it's all of them. So this blood will then percolate through these spaces called hepatic sinusoids. In the hepatic sinusoids are lined with hepatocytes, your liver cells. One of the jobs of the liver cells are to break down any toxic compounds, to purify the blood. So they will do their thing and they will purify the blood, and at the same time, they will use oxygen. So that purple blood then turns to just regular old blue blood. So what you get out the other end is oxygen poor, nutrient rich, but clean blood. So high O2 in, high nutrients in, how you get bile out. How you get oxygen poor, nutrient rich, clean blood out of the liver. So this blood is nutrient rich but dirty. This blood is nutrient rich but clean. So hopefully you can put all this stuff together and understand how the liver functions. Purifying blood. 2 ways to bring it in. One carries oxygen, one carries nutrients. One way to get it out once it's clean and deoxygenated. Connecting these two together, hepatic sinusoids. Now, a lot of these hepatocytes, they're going to be connected by these tiny little structures called bile canaliculi. So tiny little canals for the transportation of bile. All these bile canaliculi are going to join together and make a bile ductal. All the bile ductals are going to join together and make common hepatic duct, draining bile out of the liver. Okay. So just sort of putting it all together, here we have our hepatic portal system draining the intestine, stomach, spleen. All the stuff is transported to the liver where it can hook up to those hepatic lobules. where you're going to clean and purify that blood, and then it's returned via the hepatic veins. Hepatic veins are going to join to the inferior vena cava. Inferior vena cava will then take it to the right side of the heart, where it will then go to the rest of the body and feed all those hungry cells. So if we look at real microscopic histology, we can see one lobule right here, right in the middle. You have the central vein. Over here, we have a triad. So we take a zoom in at the triad. You're going to have the big one is a branch of the hepatic portal vein. The little thick-walled structure here, that is going to be a branch of the hepatic artery. And the one that has the really big nuclei, you're going to have a simple cuboidal epithelium. That's the bile ductal. So the bile ductal really stands out because it's simple cuboidal, big, dark nucleus, easy to see. The branch of the hepatic portal vein also stands out because it's so big and it's filled with blood. The third one, the small one with the thick muscular wall, branch of the hepatic artery. So these three things together are the hepatic triad. Right. Remember, don't confuse hepatic triad with the porta hepatis. Porta hepatis is artery, portal vein, and common bile duct. Right? You got lots and lots and lots of hepatic triads. So don't confuse those two terms. So the gallbladder is attached to the inferior surface of the liver. Its job is to collect and concentrate bile. So the cystic duct is going to connect the gallbladder to the common bile duct. I'll have a picture to make this a little bit more clear in a second. The pancreas, remember, has both endocrine and exocrine functions. Endocrine functions are for the production of insulin and glucagon, which control our blood sugar. The exocrine functions, this is going to make all the digestive juices that are going to help break down the food in our stomach. So let's read this next one here. Exocrine functions involve secreting digestive enzymes, which I talked about, and bicarbonate. What's bicarbonate? Right? What's up with that? Bicarbonate, we're basically talking like a component of baking soda. Well, what's baking soda? Why do we have baking soda? Well, baking soda is alkaline. It's basic. Why is the pancreas making a basic solution? Because we got to neutralize that stomach acid. Right? So remember, stomach is producing acid at pH 2.2, concentrated hydrochloric acid. That would pretty severely damage other parts of your gastrointestinal system because they don't have the protection your stomach does. So when that chyme leaves your stomach, passes through the pyloric sphincter and enters the duodenum, it's the job of the pancreas to neutralize that acid. How does it do it? By bicarbonate. Right? That's why you don't get acid burns down the rest of your GI tract, because your pancreas is going to make an alkaline solution to neutralize the stomach acid, so it's no longer an issue. So let's cut to the picture. So here is the pancreas. You can see it's drained by the main pancreatic duct, exocrine portion, and we have this little accessory pancreatic duct. Right, both of these are going to drain the pancreatic acini, making the bicarbonate, making the digestive enzymes. Where's that stuff all going? Right here. It's going into the duodenum. So remember when we talked about the duodenum, I said there was something called the major duodenal papilla, a little projection into the duodenum through which both bile and pancreatic juice are going to enter. Well, here you go. Right, there is the major duodenal papilla, connecting main pancreatic duct, bringing in bicarbonate and digestive enzymes. And here is our common bile duct, which is going to deliver bile from the liver and the gallbladder here to mix with the food. So the purpose of bile, as I mentioned, is as an emulsifier. It helps separate and break down fat so the various enzymes can break it down. Right? Pancreatic juice provides most of the enzymes that are important for this function. So here's a pancreatic islet looking at the histology. This was part of the endocrine system. All the stuff out here, the vast majority, right, this will all have ducts and things like that. This is all making pancreatic juice. So one thing I should point out here, anyone who's had their gallbladder removed, right? Or it knows anyone who's had a gallbladder removed, you have to often have an alteration of your diet. If you eat too much greasy food and you don't have enough bile, you're going to get a very upset stomach because your body will not be able to process all that fat and that grease, and it's going to just come tearing through your GI tract, and that is no fun whatsoever. So you can have your gallbladder removed. The gallbladder, because sometimes the gallbladder, the bile salts will start precipitating out, and you get what are called bile stones, and that can block the flow of bile, and that can also make you sick. So if you're prone to getting gallstones, they'll usually just go in there, cut out the gallbladder. They'll just snip this off right here and remove the gallbladder. But you'll still have bile because bile is made in the liver and it can be transported directly into the duodenum. Now, it's much more convenient to have a gallbladder because what will happen is it will be a little storage bottle for bile should you need it, right? If you eat sort of real healthy, lots of vegetables, low-fat food, the gallbladder will just sort of fill up and it will sit there. Then you go on a bender, you have like some giant, massive cheese pizza and you eat a whole bunch of fat. Then your body is going to need that bile. The gallbladder will squeeze and then it will add a bunch of extra stuff. So you can eat kind of a crazy bounce around diet if you have a gallbladder. However, if you have your gallbladder removed, if you eat that big greasy bomb meal, you're not going to be able to break down those dietary fats and it's going to make you real upset to your stomach. Okay, so let's talk about biliary apparatus. The biliary apparatus starts in those bile canaliculi in the liver and drains all the bile out of the liver and eventually connects it to the gallbladder and the duodenum. So it's going to start in the right and left lobes of the liver, make the right and left hepatic ducts. Right and left hepatic ducts drain bile out of the right and left hepatic lobes. Those two ducts join to form the common hepatic duct. The common hepatic duct is going to join with a cystic duct. The cystic duct is connected to the gallbladder, and then it's going to form the common bile duct. The common bile duct is the final stretch of the two that actually is going to reach the duodenum. And there it's going to enter through the hepatopancreatic ampulla, just another space here. So let's cut to the picture. Right and left hepatic ducts drain right and left hepatic lobes, drain bile out. When they fuse and they come together, you're going to get the common hepatic duct draining bile out of the liver. The gallbladder is drained by the cystic duct, cyst meaning cistern, which is just like an underground storage container for water. So the cystic duct merges with the common hepatic duct to form the common bile duct. So all bile flows through the common bile duct. So right, left hepatic ducts join to form common hepatic, cystic, and common hepatic join to form common bile. Common bile eventually passes through that hematopancreatic ampulla and eventually then enters into the duodenum. That's how bile and pancreatic juice get into the duodenum. And with that, we've come to the end of the digestive lecture. So normally, the way this works, I have a little extra section here talking about a not so delicate matter. However, we don't have a whole lot of time here, but I just want to make a point that the vast majority of fecal matter, 80% of the dry matter, 80% of the mass of dry matter is not undigested food. 80% of fecal matter, not including the water, is actually bacteria, right? So we have a huge number of bacterial cells that live on and inside of our body. It's called the microbiome. And this may sound kind of terrifying at first, but it's actually a very good thing because it actually helps keep us safe. Now, I'm a microbiologist at heart, so I could talk for hours about the microbiome. But I at least want to mention it here because our relationship with these organisms are so important. It may be that the next time they make an anatomy book, there's going to be a section on the microbiome, right? We really can't live without these organisms. They help keep us safe and There's a very intimate relationship between these microorganisms and our immune system and our health. And the microorganisms in your gut can actually affect mood. Believe it or not, out of the many crazy things that you can find dealing with the microbiome, they have found that there's certain species of organisms living in your gut can give you a sense of contentment and well-being in your life. So what I'm saying is that there are treatments in development. If you suffer from clinical depression, there are trials that are actually showing some pretty serious promise where you will do fecal transplants from one person to another person for a treatment of depression. Now, how all this works, I don't really want to get into, but just understand you have millions and millions and millions of different kinds of cells living inside your body, and all those cells are making all kinds of interesting compounds, which they're secreting into the environment, your intestines, and your job of your intestines is to bring this material inside your body, right? And it just so happens a lot of the stuff that these cells are making can have a profound influence on things like mood, appetite, all sorts of interesting topics we don't really have time to get into. If you guys go on to take microbiology, hopefully your micro professor will spend some time talking about it. But this is sort of a very interesting field for future research, right? And this is the last little factoid I want to leave with you. You actually have more bacterial cells on inside of you than you have cells yourself. And there are probably 10 to 100 times more genes in those bacteria than you have genes yourself. So how is that possible? Well, bacteria are much smaller than our cells. But it really raises this question, where does the human end and the bacteria begin, right? They really have a very close relationship. And why am I mentioning this? Because it can have a profound impact on health. So stay tuned, keep an eye out for that kind of stuff. If I have a chance, I might post a few videos on the subject, but it's not a subject for this test. I'm not going to talk to you about the microbiome, or not going to ask questions on the microbiome on the test. It's just a super cool subject matter, so I just wanted to share with you guys. And with that, we've come to the end of our digestive lecture. Next time, urinary. Urinary is confusing. So I'll just say now, read the book. And you're probably going to need to listen to the lecture, and you'll probably need to read the book again. So urinary tends to get a little bit complicated, and that's definitely one you want to get down now, because when you get into physio, urinary is going to blow your mind. Urinary is like crazy. You're going to learn that the urinary system and the respiratory system control blood pH. It's going to get crazy. So for right now, prep on urinary, get the structures down, and when you get into physio, it'll be much, much easier. All right, with that, I'll be quiet. Take care. See you guys next time.